Plasmids are small DNA molecules that carry foreign DNA and can replicate independently in a cell. They are considered a type of cloning vector.
The lac operon is a functional unit of DNA that contains a group of genes that are under the control of a single promoter. The lac operon works when there is lactose present and glucose is absent. The rate of transcription in the lac operon is regulated by the lac repressor and the catabolite activator protein, also known as CAP.
The lac repressor is a lactose sensor. It inhibits transcription of the lac operon by binding to the operator when lactose is not present. When it is bound, RNA polymerase cannot transcribe the operon. When lactose is present, allolactose binds to the lac repressor and changes its shape so it cannot bind to the operator. CAP is a glucose sensor. It helps RNA polymerase bind to the promoter. Without it, the rate of transcription would not be very high. CAP binds to the promoter when cAMP is attached to it. cAMP is produced in low levels of glucose. Lac operons are considered an inducible. This means that they can turn on or off the gene expression by permitting or inhibiting transcription. This allows us to change the expression of our gene of interest if we desire.
The PET vector is a specific plasmid for expression by T7 RNA polymerase. The T7 promoter system is commonly used because it is one of the strongest promoters known and produces a high yield of proteins. The plasmid vector includes a selectable marker coding for antibiotic resistance, an origin of replication, the LAC-I gene which codes for the repressor, and the transcription site with the lac operon and gene of interest. The gene is inserted after the T7 RNA promoter sequence, lac operator, and ribosome binding site. The coding sequence for the highly active T7 RNA polymerase is added to the bacterial genome where it will be transcribed by the lac operator and consequently expressed when IPTG is present. IPTG is a molecular inducer of the lac operon. Unlike its analog, allolactose, it is not metabolized in bacteria, meaning that its levels will be constant. The molecule enters the cell by active transport, and as you increase the concentration, protein expression will also increase. It works by binding to the lac repressor and turning on expression of operon and T7 RNA for transcription. Once induced, the T7 RNA polymerase will transcribe high quantities of the protein of interest under the controls of its T7 promoter.
There are many purposes for using bacteria to produce recombinant proteins. This includes their fast reproduction rate, their low cost, and their ability to be stored on ice for later use.